and other medical implants or surgical procedures that damage the skin barrier and consequently lead to HAIs. So this whole talk is about just that. So one area is medical device associated biofilm, as we saw in the catheter one earlier, where a catheter that is being placed, then surface bacteria which are normally innocuous because they're sitting on the skin surface, can now, when there is an incision because of the catheter or because of this major, when they have these in today's world, when they are doing cancer treatment and other treatment, they are putting a central venous catheter or something like that, that goes into one of the major arteries, and it is placed there for several days, maybe weeks and even months, and that thing is just stuck there, and there's a whole lot of transmission. So those things frequently develop that. So there's a lot of research on that area, and people have been looking at it. Okay, so we will also try to see some of those in small time. So there is also tissue associated biofilm, which will, for instance, where bacteria are now growing on certain cell types or tissue types as versus a medical implant. So in that case, for instance, if there are chronic wounds or mucosal membranes in the dental area, there is a lot of studies going on with biofilms. So, as I think we discussed earlier once with Dr. Sunil, there are also opportunities for, dysbiosis. Dysbiosis, the term just simply means that there is a normal equilibrium maintained, or what we believe an equilibrium is maintained within a given site, where when bacteria are growing, they're maintaining a harmony kind of a thing. However, due to different circumstances, there is a dysbiosis or a change in the number of certain kinds of bacteria, or change in the actual population dynamics. Or, like, for instance, since it's a polymicrobial film, a microbial biofilm, it can so happen that you can have different bacteria that are introduced into that area, and as a consequence, the whole system is out of balance, and that is called dysbiosis. And frequently now people are studying that, like, you might have heard of the term gut dysbiosis. Of course, that is not necessarily all biofilm, but in that case, the dysbiosis component is similar. So then these are some of the ways in which we measure bacterial biofilm growth. Uh, we have some traditional methods in the laboratory to measure how biofilms are growing, and there are numerous ways. We cannot cover all of them in one day. One is just spectrophotometry. You can put bacterial cultures in a cuvette and actually look for their adherence over time and study that. That's the simplest form. The most common version is actually doing what's called a crystal violet assay. Crystal violet is a dye that can adhere to proteins, nucleic acids, and even exopolysaccharides. So it's the simplest version. What you do is you grow a bacterial culture and you place it in usually 96 well plate format, which is where we do it. You incubate the plate for at 37 degree, which is the incubation temperature for most bacteria, and then you allow that incubation to happen at for different lengths of time, because some people try to study biofilms that are growing and maturing, and then trying to assess what is the ways to eliminate the biofilm at different stages, and also characterize them. So that's what we do. Then we remove all the media, and the bacteria that want to grow on the solid surface and form a biofilm over that time would form or have formed a biofilm. Then we wash with PBS to get rid of the non-adherent cells, fix the biofilm with air drying or using some sort of simple fixative, add crystal violet to stain the cells that are bound, and to visualize that, remove that, wash it with PBS, and then at the bottom we end up getting a layer of cells that is now stained with crystal violet. And the intensity of staining can be used qualitatively, where we are just looking at the staining and saying one is more than the other when we are trying and comparing samples. Or we can add just some uh, agent, such as 50% methanol or something like that, and actually extract the ethanol, the crystal violet from the cells. It comes into solution, and then we can spectrophotometrically get a quantitative number for how much crystal violet had bound to this. So this is one simple way to measure. And then why should we study biofilm? Well, as I discussed earlier briefly in the introduction, biofilms are associated with HAIs, as we will see subsequently. So they occur in diverse environmental niches. So, as a consequence, it is important to study them because we believe that most of the hospital acquired infections are probably biofilm related. Okay. Say they may also contribute to antimicrobial resistance because when you have polymicrobial biofilms, you can horizontally transfer genetic resistance markers from one cell to the other and things like that. So, all these have been well documented. So that's why we do this. We are also interested in what is called dormancy entering persister cells, which are cells that I did not describe in my earlier figures, but these are cells that are entering a slight phase of dormancy. And I will try to show that again in some of the slides later. These cells show a decreased transcription of their genes, and one of the genes that has been identified is GLPD. Okay, this is a specific gene that is induced in persist and it's called a persister gene. So there are also others that are probably there, and people are studying these cells because these cells are highly resistant to antimicrobial agents, and we need to understand what is going on there. So the mixed biofilms are basically biofilms where you have more than one species of bacteria interacting with each other and growing. All right. So we have frequently found that Staph aureus and Pseudomonas aeruginosa, these are the most commonly studied bacteria that are grown together, and people try to understand what is going on between the two species. So I'll move to the next slide. Fungal species also can be incorporated in a biofilm along with bacteria, and frequently you find Aspergillus species and with Pseudomonas aeruginosa in CF lung environment. CF is cystic fibrosis, where you have Pseudomonas is a predominant player in that, and so most of the time people succumb to Pseudomonas aeruginosa infection, and the dynamics is different. Sometimes you have two different bacteria uh, co-infecting and things like that. So not only bacteria but also fungi. The most chronic infections are polymicrobial. I've already talked about that, so we will not go into that. We'll go to the next slide. So common features of what microbial biofilms: adherence, presence of extracellular polymorphic substance, which includes DNA, protein, uh, exopolysaccharides, and things like that, and broken down cell material and things like that as the biofilm matures. So that whole component is called a extracellular polymeric substance or EPS. And this forms a gum like structure around it, which, as you can see, if it is surrounding all the cells, it is difficult for antibiotics to penetrate. And if they do penetrate, they form a gradient. So from the top surface of the biofilm, there is a higher concentration of antibiotics typically, and then as you go towards the bottom end, the flow of the antibiotics is changed. And what people believe with persister cells, in particular, that I just mentioned, is the fact that those cells tend to be located in areas where the antibiotic concentration is low, so they are able to survive and grow. So even if the rest of the biofilm is eradicated, those persister cells, as the name suggests, are growing. Then there is some given and defined architecture to the biofilm, and there is also a viscoelasticity, which basically means that the biofilm is stuck to the solid substrate, and you can study that by using, uh, you know, flow reactors and stuff like that, where you are trying to move media over that biofilm and studying the dynamics of how fast it is released or how fast it grows and things like that. So you can do all of those studies. We are not able to talk about all of that here. So the life cycle, diffusible cell signals, altered microbial environment, microenvironment formation, which I briefly discussed, because when you're talking a flow of nutrients and flow of antibiotics from the outside, there are gradients that are created within the within the biofilm per se. As a consequence, different cells are exposed to different levels of nutrients, pH, and stuff like that. So you have these dormant or slow growing subpopulations, which is what persistent cells are, and small colony variants are. So going to what are nosocomial infections? Nosocomial infections are just infections that occur in a hospital. So typically people believe, for people who are naive, they believe that when I go to a hospital, I will get cured. However, prolonged stay in a hospital can result in patients acquiring infections from procedures conducted on them. So when they do surgery or they do other things, they insert probes or things like that, or they're being examined by stethoscopes. Even stethoscopes can carry bacteria and things like that. All those things can transmit bacteria to the person and cause some level of illness. And when that is done, that person's recovery rate becomes low. And so nosocomial infections tend to be infections that delay departure from the hospital. So if you're admitted to the hospital for 1 day or 2 days, it may happen that you may develop fever after 2 days. In fact, that is the very basis for nosocomial infections. People say it's a nosocomial infection when a person enters a hospital perfectly normal, or normal from the perspective of a fever or something else, but he has some other malady that has come for cure. But after 48 hours, he develops some infection. Then that infection is called a nosocomial infection. So, how do these things can happen? Procedures such as surgical incisions, insertion of probes and catheters, indwelling catheters, like we talked about the major. I think I have an image of that. If not, I can supply that to you. These can cause HAI, or hospital acquired infections, or nosocomial infections. HAI and nosocomial infections are one and the same. Guests coming in to visit the patient can bring in infection. So when you have people visiting during visiting hours, guests come in, they can bring infections from the outside. Hospital cafeteria can bring foodborne pathogens. So if the hospital cafeteria is not exactly clean, or because it is overcrowded, as it sometimes happens everywhere, they can bring this thing. And then when there's leftover food, animals and other insects and things like that come in, they also can contribute. Hospital environment derived infections from pathogens occupying environmental niches. This is what is the crux of the topic here, commonly found in hospitals. So, what are these areas that we are talking about? We'll discuss some of those today by going through the entire set of series. Prevention. So, there's usually in the hospitals to negate the levels or reduce the numbers of nosocomial infections, there is an infection control officer who maintains strict control, and his job is to instruct the HCW, or healthcare workers, to determine or to find out what is the status in terms of how well are the nurses trained, how well are the other people trained in the procedures of hand washing and all these other parameters in the hospital. Even the hospital design scenarios, which we can't go into, are carefully monitored so that hospitals can maintain a strict control. And hopefully today in the talk, you can see some of that. So here in the second sentence, these are hospital-acquired. Nosocomial infections are those that are acquired by a patient within 48 to 72 hours or 3 days of admission into the hospital or medical care unit. So when the patient comes in, he may not have a temperature, but he may have a broken leg. So he's come to fix his broken leg. He gets admitted to the hospital for a day or two, and within 2 or 3 days, he develops a fever. Then in that case, that fever is probably a nosocomial infection that he acquired in the hospital. They may occur in different areas of healthcare delivery, such as hospitals, long term care facilities, ambulatory settings, and may also appear after discharge. So, a patient is staying there for 2 days and he is released, he may then go home and develop a infection. Just to give you a simple example, I was in for an operation in US, and I was being operated for gallbladder stones, and they removed my gallbladder, and then they said you will be released the next day, but the next day I developed a fever, and the fever did not go for a week. So I was stuck in the hospital for a week, and I got a very big bill, which is part of the way how these things happen. So sometimes people have low resistance in the patients to infection because they're already ill. So when they come into the hospital, they have other underlying situations or disease conditions such as diabetes, cancer, mental disorder, immunosuppression from some other thing, or they're coming in for an organ transplant where they undergo immune suppression. So then these kind of scenarios play out in nosocomial infections even more, because here the patients are actually having a lower resistance to than a normal person. The natural mechanisms of the body surface may be bypassed by injury to skin or mucous membranes. And then you can get other things like burn wounds or surgical incisions or procedures. Then bed sores happen when people are not moved frequently. If they're not moved for more than, uh, you know, 15, 20 hours, then they develop bed sores and mites from linen and healthcare workers and other medical personnel. So we'll look at some of those things today. Contaminated environmental sites. So these are different bacteria that can be present in environmental sites within the hospital. So where they found like gram-positive cocci are frequently found in air and dust and on surfaces. So pathogenic members of this group are of human origin and they may survive for a number of days in dry situations. In fact, we will discuss Legionella and Pneumophila, and Legionella Pneumophila strains have been known to be existing for up to 17 years in certain areas. So that shows how long things can persist and what weird environments they can persist in. So there are spore-bearing anaerobes that may be introduced into the hospital from outside air or unsterilized object, or they may be released into the hospital environment from dried feces or wound exudate. People may have come with a wound or something like that. All these things, even from the OPD, things can come in to the other wards and can transmit to the ICU. So we can't go into that, but for my hospital acquired course, I did prepare all of those, and I have lived in a hospital environment when I was doing my PhD work. I was in the middle of the medical center. So I'll give you a little bit of that also. Drug resistance of endemic microbes. The large proportion of hospital patients receive antimicrobial drugs as it is when they enter, and now things have gotten much more sophisticated, but back when I was working associated with the hospital, they were not that sophisticated. But some normal body flora are sensitive to the drug and they can get suppressed, but resistant strains can then appear within the hospital setting itself. And I don't know what I did there anyway. So when the person receives an antibiotic, it's very common that you can develop some antimicrobial resistant strains within the hospital setting in itself. So you have to be careful of those. And these are the four common healthcare associated infections. This is what I was talking about, center line associated bloodstream infections or CLABSI. In this case, what happens is you have this huge catheter that is placed somewhere here in the upper region of your chest, and it is entering one of the main arteries, and it is kept there for a long time. I can't go into the medical part of it because I'm not trained into that area, but I have, of course, witnessed this with my own family members, my own self as well. And in this case, these things like center line associated ones are the ones where people are being either are completely immobile and sometimes they're in coma or whatever. They are being fed continuously drugs and other things that are going through those lines. Then there are catheter associated urinary tract infections, surgical site infections, and ventilator associated pneumonia. Um, so all of these are the four major categories of hospital acquired infections. Um the causative can be anything from bacteria, fungi to viruses, and we will see some of those in today's talk. So pneumonia is hospital-acquired pneumonia is just pneumonia that develops after 48 hours of admission. So ventilator-associated pneumonia develops 48 hours after an endotracheal tube intubation, which is where they put this tube into your mouth and your mouth remains open, and this tube is placed and you're assisted in breathing. So there is a ventilator that forces air, and it's for people who are debilitated and have pneumonia, and they're forcing more air into the body. So approximately 5 to 15% of mechanically ventilated patients develop VAP, which means basically when out of 100, up to 5 to 15% of those will actually develop pneumonia as a result of this very thing. So patients develop this as a result of aspiration and inhalation of contaminated aerosols. So when in the hospital environment, even though there are filters placed in there, there is your mouth is continuously open, and this tube is placed in there, and so bacteria from the air, surrounding air, can go in. And I can give you horror stories of hospitals because I've seen them, and I hope I never have to go through this. So or anybody has to go through this. So it is very difficult. Placement of indwelling urinary catheter, meaning a catheter that has been placed inside. During treatment, 60% of critically aged ill patients will receive a urinary catheter. 20% of the patients in medical and surgical units, and 5 to 22% of residents in long-term care facilities will get a catheter at one point or the other. I also, because I was unable to pass urine, they had placed a catheter after day third into me. So that was the first time and only time I ever got a catheter. So now, once there is an indwelling catheter, there is a good chance that there is bacterial growth uh, in the urine, which is called bacteriuria, and then because there when there is this insertion, if it is long term, there is a catheterization that is greater than 28 days, results in continuous this thing and symptomatic urinary tract infection. So this is very very common, and I've also faced it. And here's just some examples of different catheters. And you can see here there is an example of a catheter which has been damaged because of biofilm formation. And because the formation of the biofilm, the lumen of the catheter has been damaged, and as a consequence of it, the catheter becomes non-functional. So they have to remove the catheter, which is one problem. That means the device fails. And then the second problem is that because of that bacterial infection per se, the person develops an infection, so that becomes systemic. So here is an example of some that are treated with different drugs, and you can see that they are less this thing. There is no occlusion. Then in the next slide, I'll not go through the slide completely, but you can see because of formation of the biofilm, this is a normal tube, and there here you can see damage to it that allows it to break. So this is a case of where device fails because of the formation of the biofilm. So, what is an environment? Let's shift focus now to. We have talked about what biofilms are, and we've talked about some basic steps of some concepts of HAI. So, let's shift gear and talk about what is an environmental niche. What is it? So, in ecology, a niche is an area or an environment within a very focused environment where a bacterial species or any other species can grow happily and is able to grow because it can relate to that environment, and it has been tuned to live in that kind of environment. Like when we talk of animals like tigers and lions and things like that, we talk of lions being animals that live in an open environment, while tigers living singularly tend to live in a thick forested area because they like to ambush prey. So that is a ecological niche that the tiger occupies as versus the lion, which is totally different. It is living in a large safari like area, so that is totally different. So here a species carves out a niche for itself where it is able to grow. And why is it relevant? I mean, why am I talking about environmental niche? Because in a hospital there are different environmental niches where different bacteria will grow in certain things based on their physiology. So some things that we will probably learn today from the talk it will be fascinating for that reason. Why should we consider these? So hospital instrument probes, for instance, stethoscopes, ultrasound probe, thermometer, glucose meter, keyboards of instruments, X-ray machines, ultrasound, and other glucose monitors. If you have ever visited an hospital and have had the unfortunate event where you had to be hospitalized for some reason, you will find that you have to be tested by X-ray or some other machine comes to test you for ultrasound. They're not always perfectly clean because you are tested, then somebody else goes and he is tested. So there is a lot of movement of patients, patients who are ill, critically ill and chronically ill, who may be carrying an infection or may have acquired an infection. And so through their mobility, a whole dynamic is established in the hospital where these different materials can be, tested. And I think we should test them. Air conditioning towers, I'll discuss Legionella. You can see that Legionella is a classic of stagnant water in air conditioning towers. If you notice in the olden days, there used to be water towers where the water was forced to circulate and its temperature would be reduced and then was used in air conditioning. Um, those cases, there were several instances of Legionella contamination in those waters. So countertops and surfaces in rooms and ICU, they can get infected where biofilms are actually growing because you can have that. In cafeteria and insects, people have isolated from cockroaches many bacterial species that are pathogenic. So that's just an example. And cockroaches are very common in cafeterias and things like that. Water fountains and water reservoirs, including distilled water. And one of the things that is not pointed out here are solutions that are used like salines and other antibiotic, not antibiotic probably because they don't get that infected, but other solutions, medicines that are being used for people, they can frequently be infected with bacteria, and those bacteria are sitting latent there unless they are then put into somebody, and things like that happen. So bathrooms, sewer systems, and drainage, fruits and vegetables, as we shall see. Wheelchairs, which is a very interesting concept. Some person did a very interesting study with wheelchairs and demonstrated how wheelchairs, if you have never noticed, and had to sit in a wheelchair in a hospital. That wheelchair is there for you for only 5 minutes. You're taken to the hospital inside, and then somebody else uses that wheelchair. So you are placing your hands, and I think if we can get to that slide, I'll show you. So this is a focus on slide which depicts different parts of a hospital bed and an open ward. And as you can see, there are so many different components here that can actually harbor bacteria or viruses. And so we will talk about that. This is the air conditioning tower where you have this water circulating that cools the water, and these are large reservoirs of water that can sometimes get infected. This, interestingly, is a picture of me sitting in front of the University of Tennessee, UT. You can see here, University of Tennessee Bold Hospital. This is right next to where I used to work because it's a medical complex. So in US, all the medical hospitals are located in what is called the downtown area or midtown area, so that all the people from different areas can come there and get treated. That's how the hospital system. And so it has about 10 or 15 hospitals ranging from different varieties of hospitals, hospitals dealing with cancer, hospital dealing with something else. So this is the university's teaching hospital, UT Bold, which where I was, like, this is in 1993 or 94. So it's an old picture of me sitting there and taking a picture. But you can see the outside environment of a hospital is fairly good. But as you move inside, uh, the hospital can actually be not a very good place to be. So these are some sources of nosocomial infection: direct, that is staff to patient, patient to visitor, patient to patient, and then indirect, which is aerosols. Like if you're breathing out, viral particles, even bacterial particles have been shown to be transmitted through aerosols, through a vector, that is through an insect bite like malaria, and some other like yellow fever, that is also there. Use of contaminated medical instruments, which is basically fomitis based transmission. So you have this. Then spread of various pathogens. So here we won't go through this list because you have these different bacteria can be spread through air, and then it describes this complication slide. So I encourage anybody who wants to keep these slides can keep these slides to, if they're interested in this kind of research or area. Then this is contaminated aerosols, then electrical or other appliances or air conditioner. In this case, legionella is one of the classic examples. um, so here we can see how different pathogens are spread. Here there is a clear demarcation of how direct contact happens. So you have contaminated air by an infectious agent. So maybe you are a visitor or something, and you walk to a patient or a friend who is a patient, and when you come in contact or come close to that person, you can get infected because some healthcare worker, maybe not your friend or patient, but a healthcare worker may be carrying a bacterial infection or a viral infection, and that can be transmitted to you. So then exposure with another patient, exposure through healthcare worker, patient visitors in a hospital, contaminated water. All of these are direct contact kind of things where they can lead to this. Through medical instrument, contact in contaminated inanimate object, like you're touching a doorknob or something like that. So this can happen. I mean, all of these slides have been prepared very carefully because for each of these events, I've gone back and done some research. So I have papers that discuss that. So then this is airborne disease caused by virus, different viral viruses that can actually spread diseases, and what kind of disease it is. So you have to be mindful of those diseases. And here also you have airborne nosocomial infection. So within the hospital, basically it's a repeat of the same slide. So I'll skip that. And I wanted to go to this slide because this describes Legionella. So, hospital air, water and air. On July 21st, 1976, the American Legion opened its annual 3-day convention at a hotel. This is the hotel, and they didn't realize when they did this that they had this convention and people came there. They attended the convention. The hotel was air conditioned, and when they left the hospital, some people developed this Legionnaire's disease, which was at that time not known as Legionnaire's disease. It's basically a pneumonia like disease, and these people acquired that infection. Some people About 29 people died, and the Legionella bacterium in 1977 was then finally identified. And here you can see a couple of people working to try and identify the bacteria. And what it was found was that it spread through the cooling towers in the air conditioning system of this hotel. So at that time, way back in 1976, they did not know about this possibility. So this is how, even though this is a hotel, it's different from a hospital. It's a confined environment, and air conditioning system ducts and all that can transmit bacteria. So Legionella is interesting because it causes Legionnaire's disease, which is a serious type of pneumonia. It's a lung infection. People can get sick when they breathe small droplets of water or accidentally swallow water containing Legionella into the lung. And these are some images of this gram-negative bacterium. And so then we talk about how many transplant units and all there, and there are some statistics here of the thing. Because there are so many slides, I will not go through all the components of the slide. So here there is a survey that shows that only 5% of the healthcare facilities have developed and implemented a waterborne pathogen risk management plan for building water systems. So the water systems are not that still well developed. Even in the USA, and many viruses can be transmitted also through water, and so can protozoans and things like that. So, who are the high risk people for these kind of infections? These are people who are in intensive care ward patients, neonates. Neonates are newborn babies, and patients with HIV and AIDS, cystic fibrosis, and undergoing, complicated procedures like renal dialysis and transplantation and things like that, and burn treatment. These are the people who are high risk to infection from these, because not only are they there in the hospital for long periods of time, they have all these implants and things like that that are going on in their body. So in Germany, they did a simple experiment where they used a zero point 0.2 micron point of use tap filter in three intensive care units, and they showed that when the point of the filter was placed at the tap, and they were using water that was filtered through a zero point 0.2 micron filter, the presence of Legionella in 251 samples was zero. However, in the other cases where they recovered Legionella in all these other samples when it was unfiltered. So that shows that by just placing a filtered tap water system in a hospital, you can reduce this. Now, I won't go into that, but most hospitals are develop an infection control plan, and they use all these kind of materials, and it is then based on a budget. Also, because budgets determine how much expenditure they can do, and how many healthcare workers and how many nurses they can train, and all of these factors go into control of nosocomial infections. So Legionella biofilms, here you can actually see the interesting concept about Legionella is that it actually lives in association with amoeba and things like that, and is actually internalized by amoeba. So here you can see a, 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 a species of amoeba that has ingested Legionella that is expressing green fluorescent protein. Okay, so when green fluorescent protein is being expressed, it fluoresces, and you can see internalized bacteria in this image of amoeba that has ingested it. And there is a lot of studies with using different forms of amoeba, and amoeba that are heat tolerant and things like that. And when they've used heat tolerant amoeba, they can put Legionella there and mix the two together and show that now the Legionella can survive. Um, Move little fast. Okay, sure, we can move little fast. So anyway, this is how we can talk about this. So there are few main ways to limit nosocomial spread, and that is treatment of biofilms with antimicrobial agents and a lot of research, development of novel antimicrobial surfaces that resist biofilm growth, identification of ways of spread of bacteria and viruses through research, institute training of healthcare workers to control nosocomial spread, and establishment of global networks to study, monitor, and control AMR and HAI. So when you have these different diseases that are appearing, we can do that. So here, the UK's National Health Service has, not go through this slide, because focus is biofilm structure and pathogenesis and therapy. So biofilm is, because it's the key. A lot of treatment options are there where all these different phases of biofilm are being tested. Some examples are there on the slides where people are using drugs and then they are also altering the structure of the implants or or the material, so the topography, so that and they're doing chemical modification. They're doing smart surfaces where the antibiotic is released in response to pH alteration or temperature, or by addition of some other encapsulated drugs that has been done. Oops, sorry. So if I go to the next slide, here you can see they're using antibacterial nanoparticles and all these slides. All these slides are available, so I will not go through these in detail because, as in the interest of time. Then there is physical removal by using ultrasonic waves or water jets and things like that. Then there is DTT debridement that is also done. So all of these techniques can be done, and then these are just ways in which different bacteria attach to the surface. People have used different mutants to study what proteins are involved in association with the surface. People are doing transcriptomics on different stages of biofilm to determine what genes are expressed in different stages of biofilm. Here is an example of using bacteria that are expressing red cherry, and then they are using different catheters that are coated with different compounds, and then they are testing the growth of bacteria by looking at red cherry fluorescence. So this is an example. We can see that clearly. So I'll skip some slides to this slide where they are using, biofilm eradication strategies for titanium implants. They're modifying the surface of the titanium at nanoscale by using spear-like projections on the. This is at nanoscale, nanometer scale, as versus pocket type and polished titanium, and then they're comparing which of these surfaces adheres bacteria the best. So they've discovered that when you have spear type this thing, the because the spear projections are so close together, the bacteria are unable to attach as well as the other two surfaces. And then they have also used things like rhamnolipid, which is a surfactant, and they've shown that as you add increasing amounts of rhamnolipid and you look at the biofilm formation, it is altered. So these are the graphs for that. um, I think I'll go to this slide directly where they talk about using different combinations of therapy like drugs, surface structure, photodynamic treatment, and things like that to look at the biofilm formation and lack thereof. So then this I'll skip few more slides because here there is people are frequently using this as a way to establish how many biofilms, how much study is being done on biofilms over the years. So this is starting in 1970s. There's hardly any biofilm work, and now there is an exponential growth in biofilm studies. uh, and so and then these are the different parameters that they're looking at, like dental wound and cystic fibrosis based biofilms and studies. So this is a reflection of the number of papers that are coming out in biofilms. And this is the next slide is talking about the use of different clinical trials and the areas where the clinical trials are going on, as versus say USA versus Africa versus Russia or Europe. You can see there is a lot of intense study in Europe and America, and I don't know what this color is for, but in India also there is a lot of research now in this area. So this is a summary slide. Basically, we should focus on generating awareness and education among students and their peers, incorporate awareness for biofilms on education, and create global networks, which I'll give you an example of here. Now, this next slide, I'll not talk about it, but when we talk of nosocomial infections, one thing that I uh, alluded to right at the beginning was the fact that people who are coming into hospitals actually are having some illness because of which they're coming in. So they are not immunocompromised, but their ability to fight infection may be lower. In this case, we are looking at a more complex scenario or a more debilitating scenario where we are talking about HIV positive patients. So patients with HIV positive patients, what is happening? Not only I look at it in a one way where we're saying that when HIV positive patients are coming, especially in places like Africa where people don't even know their HIV status, and they're coming into hospitals. So it is a very frightening scenario because there are people who are HIV positive and they do not have proper medication and their heart therapy is not going on. So those people are coming into hospital and being exposed to TB and things like that. So you can imagine what the scenario is in those countries. Then these are some of these aspects where we are talking of different bacterial infections and how people who are actually having very good control on their HIV status, are able to manage that. Then impact of malaria and HIV co-infection. So there are co-infections. So when you, if you are HIV positive, or if you are malaria positive and you acquire HIV, something like that, but more on the lines of being HIV positive and then getting malaria, the outcomes are not that good. So similarly, tuberculosis is the major killer for HIV positive patients, and it's endemic in areas of Africa, which is one. So this is one of the areas. Then some novel microbes. So here we have different bacteria that are new. So Stenotrophomonas maltophilia, even I can't pronounce it, but these are new bacteria that are coming up that are causing nosocomial infections, and you can see uh, uh, they're found in hand washing soap, irrigating solutions, sink drains, faucets, and other areas. So all these different areas you can find them, and people are discovering new bacteria because as the genomics is there, we're easily able to quantitate bacteria and find out what they're doing. Then there is another. This is Fingomonas species. Um, then I'm just going through Ralstonia. This is another one that has come. Then not only that, but what's more interesting is there are some amoeba-like bacteria, like acanthamoeba, that causes eye infection. It's shown to harbor many viruses and bacteria as well. So, when you're looking at the dynamics of HAI, it's not restricted only to bacteria and viruses, but the fact that you can have amoeba and other things coming in through contaminated water that are actually harboring very cleverly bacteria and viruses. So, the dynamics is much more complex than we originally thought. So algal blooms is another thing. So algal blooms happen and they create toxins. I'm just going to go through these slides, and why they are damaging, because they cause respiratory problems in people. So people who are already ill in the hospital, they can suffer from it more. Then there are different toxins present in algal blooms and things like that, and they cause different illnesses. So they are also included. So when I started teaching this course on HAI, I had these incorporated. Then global surveillance. Now there is a global surveillance for influenza, uh, that is called GISRS, Global Influenza Surveillance Network. Sorry, it's now called the Global Influenza Surveillance and Response System, GISRS. And in this case, there's a laboratory based surveillance system for influenza. As you know, influenza, there's a whole family of influenza viruses, and people in America are always worrying about what form of influenza is going to predominate because they need to make vaccines for that kind of family of viruses. So we need to study that. Here, there are these global. This map just depicts different uh, laboratories that are existing. Some are smaller, some are bigger, and they're all in a response network. So whenever there is a epidemic of or small outbreak of influenza in one area, it can be relayed to all the other environments so that people know what kind of disease is there. So there is International Severe Acute Respiratory and Emerging Infection Consortium, or ICRRIC. This is a worldwide initiative. It talks about different bacteria, different viral components, and so it is even more uh, broad scope than the other one. So then there are challenges. The challenges are in the obtaining different viruses and collecting the samples and how good the testing procedures are and things like that. Um, I pulled these slides from my HAI course. So here I've just talked about the biofilm array. We won't go into it because we don't have time. I've already taken 1 hour, close to 1 hour. So you can see there are multiplex PCR based ways to determine, and in this case, they can determine about 20 different viral or bacterial strains collectively in one sachet kind of thing. You can add your sample and then you can find out what kind of infection there is, and it's all fully automated. I encourage you to take these slides at any point and look at this. So there are other things, and this is the wheelchair one that I wanted to show briefly. So if you have a wheelchair, these are the contact points that have been determined to be areas where bacteria and viruses can be most grown. And one of the very beautiful experiments that they did. This is the a diagram, kind of a like one of those earlier phase uh, route map kind of thing where they're determining how a wheelchair travels from one part of the hospital to the other, because that's very relevant. Because if you are in a critical care hospital, you may have people with infection, and then that the wheelchair that travels through across, how it goes across, they've done these determination for a bunch of wheelchairs. So it's a very interesting concept showing how hospital acquired infections can actually spread within a hospital, and by studying these kind of things we can come up with better preparations and also better disinfection of the things. So one more thing that I want to talk about is just the fact that diabetes is a major public health issue, and people have now claiming that there is some correlation to diabetes and alterations in the immune response. So people with chronic diabetes or very high sugar tend to have debilitating circumstances. Not only that, but what's more interesting is that when people are undergoing diabetes testing, the lancing and fingerprinting and insulin injection is actually also causing hospital acquired infections. Because if you go to a hospital and you're admitted and you tell the person that I am diabetic, they will first do a finger prick, get your blood sample, and do all these other things, and it is leading to some instances of hepatitis B virus and other viral infections as well. So just because there is a pandemic, not a pandemic, but an increased incidence of diabetic people, it is leading to another form of acquiring HAI. So majority of these can be done by cleaning the samples properly and educating the healthcare professionals. So this is a slide on proper hand washing. I won't go through the slide, but I'm pretty sure most of the people here would know exactly how to do hand washing procedures very well. And with that, I will come. As you can see, there's a list of ways to get nosocomial infection. It's huge. So there's an ever increasing way to get infected with morbidities riding. What I didn't talk about is, people having multiple comorbidities, like they're having mental disorders, they're having diabetes, they're having obesity, and then they're having respiratory problems. And so, in that circumstances, the management of HAIs becomes even more challenging. So, with that, I'd like to end my talk, and I thank you for your attention. I'm sorry I went through like over 100 slides in this time, and I skipped through a lot of slides. But I thought
Thank you very much, Dr. Sonal Motra. You covered all the topic very well, and it's it's a big topic even. So, in
yeah,
conclusion, I just want to add something that it's the nosocomial infection require a multifaceted, or you can say it's a multifaceted approach in involving like rigorous hygiene practices, effective infection control measures, and continuous education for healthcare provider and professionals so
yeah,
that
and also
yeah.
diagnostics.
So
Diagnostic kit data, because earlier something which I didn't focus on was POCT, which is point
mhm.
of care technologies. So earlier, samples were sent. Now the samples are being analyzed for infection at a much faster rate. And
So
like my
Dr.
mother
there are
succumbed to
one two
Pseudomonas
questions.
infection,
So one
so
two
she had
questions are
Pseudomonas
there
infection.
for you that they ask is, sir, which media we use for isolating the bacteria from air conditioner like air.
Uh, I have not isolated from an air conditioner, but usually you will use, like, say, if you wanted to see if it has Pseudomonas, you would use
Mhm.
a selective media. So for Pseudomonas, the easiest selective media is Pseudomonas isolation agar, which has some levels of triclosan in it, which is
Mhm.
a detergent. So when you put Pseudomonas on it, it grows happily on triclosan, no problem at all. So you can use that. And so if you want to see whether your air conditioning sample has that, I don't know what media they use for Legionella, but for Pseudomonas, you use that. For staph, on the other hand, you can use mannitol salt agar. That's a classic gold standard for staph aureus.
So thank you very much, Dr. Sonal Matra, on behalf of entire team of MBSI, especially that Dr. Soranjit Singh. They had also another meeting like at 6:15. So they were telling me that, okay, we'll bind up. Then
Yeah, yeah.
he has to join another meeting. And Prakash Halami sir also asked you one questions. We want to like reply like to what extent biofilm reduced microorganism gains resistance to antibiotics? Like at what extent like?
Can you just repeat that question one more time?
That to what extent biofilm produced microorganism gains resistance to antibiotic?
Okay, that's a good question. Again, it depends on the bacteria working with. Some bacteria show very high presence of this thing. And antibiotic resistance per se is not limited to, like if you're measuring it in the environment in a laboratory, it's not limited to just the bacteria per se, but presence of other components, like for instance, the exopolysaccharide. Okay. So when you have extracellular polysaccharides and other materials, they prevent inflow of this thing. Now, what we have done in the laboratory, we have used mutants. So we have mutants that are not making alginate, they're not making other polysaccharides. So in that case, their antimicrobial profile is different. They are resistant. Now, the persister cells that I talked about briefly, those persister cells actually have a higher resistance to antimicrobial agents, but it is not exactly known why. Okay, because it's not through the classical pattern like they're expressing efflux pumps or something like that. It is not that, it is some other mechanism, because they're growing so slowly. So I was just trying to research on that, and the latest that I know is that they're growing slowly, and we need to understand why. Because they're growing slowly, they are actually showing resistance to antimicrobial agents in a different way. The topic that I did not cover is where people are trying to use not only antimicrobial agents, but they're looking at synergism between different ways. So they're looking nanoparticles, or they're looking at other components and seeing, by adding different antibiotics, what kind of synergism can we get. They are also using plant extracts and other components derived from plant antimicrobial compounds that they are testing along with routine other things. So in that case, what they're doing is they're reducing the concentration of the drug, so that there is not too many antibiotics put into the system, but they're adding another component, which is a plant-based derivative. Like bicalin is one example. They can use something else. In that case, they're then looking, is there a synergism, and can we reduce the antibiotic concentration and get the same level of killing? So they're doing those kind of things.
Thank you, sir. So, any other from the like audience want to ask any questions? So, Dr. Mlotra is here to answer your all questions related to the topic. So, you can unmute and you can ask directly if anybody wants to ask.
Okay, I think Dr. Sunil, if there is no more question, then you can please say the vote of thanks.
Okay, thank you. Thank you very much, sir, for your time. And thank you, Dr. Sonal Mrotra, for your this enlightened talk on the biofilm and all the hospital nosocomial infections. What are the role of these biofilm? And we hope that, effective control of infections control. And if we all like working in the hospital setups, they are like microbiology team and microbiologist, if they pay an intention towards these particular measures which you have already discussed, and there are many studies related to that. So if we'll
Yeah,
pay attention towards these issues, then definitely we are going to save our future generations from the drug resistance, which is going to be increased now,
yeah,
because there are some
yeah,
data that up to 2030 where more than like 4 to 5 million people die without antibiotics, because the antibiotics they are getting resistance. so
yeah.
Dr.
Very
Swaranjit
true.
sir is also making very, 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 I think, huge round of applause for Swaranjit sir. They are arranging the people around the world, the microbiologist, working tirelessly for the MBSI from the Chandigarh area, from the Tri City. So, sir is the only person who is working day and night and arranging the speakers for the microbiologist Society India. So thank you, sir. You are with us for with the team of MBSI, and we are getting this opportunity to interact with the scientists, the big people. So otherwise, if we are going to meet these people, then it will be a very difficult
Well, that's why
journey.
with Dr. Swaranjit, I had discussed the idea of a biofilm bank. That if you have a biofilm bank of different clinical isolates, you can try using a high throughput system, which I am trying to develop, to
Yes.
study different antimicrobial compounds and combinations. And then you can then extend that to a service providing situation where any new novel microbe that is drug resistant, you can test within a given time frame using a parameter of set parameter of antimicrobial treatments, and see what we are getting, what result are we getting. Is
Dr.
it?
Sonal, we will discuss in details about
Yeah, please.
this.
I know you have another meeting.
In some
Yeah,
other meeting, we will
sure.
discuss in details. Okay.
Yeah. Thank you
Thank
so much.
thank you.
Thank you so much
Thank
for
you.
the opportunity. Take
So
care.
thank
Thank
you
you.
everyone for your time. Thank you, sir. Thank you, Dr. Sam, for your time.
Anji.
Thank you very
Take
much
care.
to all.
And
Thank
thank
you,
you,
audience,
Dr.
for
Sunil.
your time.
Yeah.
Thank you, Dr. Sunil, for the nice coordination of the meeting, the vote of
Yeah.
thanks, and the introduction. Thank you very
Yeah.
much.
Thank you. Thank you so much.